See it?
Hello, everyone. Thank you for joining and welcome to today's webinar, NASH Animal Models and Their Clinical Relevance, Quantitative Histopathology of Hepatic Fibrosis. We look forward to an engaging discussion. I am Daja Harold, Senior Marketing Manager with WuXi AppTec, and I will be moderating today's webinar. A few housekeeping bits. Please know you may adjust the size of your viewing screen by clicking the square in the upper right-hand corner. We'd love to hear from you and encourage you to ask questions via the Q&A tab at the bottom of the screen. Please type your questions there, and we will be happy to answer them at the end of the presentation. As your moderator, I will pose as many as time permits. Please know a recording of this webinar will be available within 2 days via your registration link. I'd like to share a few words about WuXi AppTec before we begin. WuXi AppTec provides a broad portfolio of R&D and manufacturing services that enable companies in the pharmaceutical, biotech, and medical device industries worldwide to advance discoveries and deliver groundbreaking treatments to patients. As an innovation-driven and customer-focused company, WuXi AppTec helps our partners improve the productivity of advancing healthcare products through cost-effective and efficient solutions. With industry-leading capabilities such as R&D and manufacturing for small molecule drugs, cell and gene therapies, and testing for medical devices, WuXi AppTec's open access platform is enabling more than 3,900 collaborators from over 30 countries to improve the health of those in need and to realize our vision that every drug can be made and every disease can be treated. Our first presenter today is Deming Zhu. Deming Zhu, Executive Director, is currently the head of Department of In Vivo Pharmacology, Biology Business Unit, RSD. Prior to joining WuXi in 2010, he had 15-plus years R&D experience in pharmaceutical and biotech industries and academia. His current research interests include in vivo pharmacology of Nash and hepatic fibrosis, infectious diseases, respiratory and neurodegenerative diseases, aging, microbiome, and diseases, etc. The WuXi teams under his management have contributed to R&D and preclinical programs that advance 20-plus compounds to preclinical candidates and regulatory filing, several of which are in clinical trials. Our additional presenter today is Gideon Ho. Gideon Ho is the co-founder and CEO of HistoIndex, a Singapore-based spin-off from A-Star and the provider of the world's first fully automated quantitative stain-free imaging system and AI-based digital pathology platform on the accurate assessment of fibrosis and other diseases. He is responsible for the partnership with multiple pharmaceutical and biotech companies and oversees the application of quantitative histopathology to NASH clinical trials and the collaborations with both industry and academia on preclinical studies of various fibrosis in animal models. For those of you just joining us, welcome again to today's webinar, NASH Animal Models and Their Clinical Relevance, Quantitative Histopathology of Hepatic Fibrosis. And with that, I'll turn over to the first presenter, Demi.
Thank you very much, Deja, for the generous introduction. Thank you, the audience, for taking the time to join us for this 1-hour presentation on quantitative histopathology and how it's used to address animal model of NASH and in particular hepatic fibrosis. So basically, this is a collaboration between WuXi and HistoIndex, and we're going to divide the presentation into 3 parts. In the first part, I will cover the pathophysiology of NASH and the questions we try to address in the animal model. And Dr. Ho is going to present the HistoIndex AI-based stain-free digital pathology technology. And follow that, I will describe how this technology is used to quantitatively analyze fibrosis and address the question of clinical relevance of animal model. So our focus of this slide, this presentation will be on the clinical relevance of such model. So trying to, in a way, to weed out, I would say, the non-hacker equivalent of animal models from this. As you can see, there's a saying, statistics, all models are wrong, and some are more useful. So in pharmacology, none of the models are right, and some are more irrelevant. We want to see how we can use the technology to weed out the irrelevant. So this slide actually represents the NASH clinical landscape, and this is a heroic battlefield, if you will, and because with mixed outcomes and a few unfortunate outcomes. However, the trial is still ongoing, and we're seeing encouraging trials results in phase 2. For example, 2 highlighted in at the bottom left of this slide. So I want to start my presentation with the basics. Okay, so the NASH pathophysiology. So the transition from an FLD to NASH, has been thought as a cumulative process with hepatic steatosis as the primary triggers, and then involving hepatic inflammation, cell damage, fibrosis, and indeed. This mechanistic understanding of NASH is actually reflected in the therapeutic options being pursued in the clinical trial. The biology underlying these therapeutic options, in turn, reinforces the pathophysiology as we understand. So this reinforcement trap, I think, may have contributed to the difficult encountered in the clinical trial, as shown in the previous slide. So in a most recent review, it's been suggested that the transition from an FLD to NASH is actually underlined by a metamorphosis, if I may use the words metamorphosis, of lipid, and compounded by pathogen-associated molecular patterns from the GI tract. So if you look at the non-esterized fatty acids, unsaturated fatty acids in the liver, 60% of them actually come from adipose tissues, and 25% are from de novo lipogenesis in situ, and was only 15% from dietary intake. So as enough LD progresses, the unsaturated fatty acid in adipose tissues undergo transformation. I mean, I lose the term liberally transformation, not in the sense of cancer transformation. So producing a so-called toxic lipid species, and together with other pro-inflammatory signals. And these changes, not only initiate hepatic inflammation, but also remodel the liver GI tract interaction such that the gut microbiome yields the so-called pathogen-associated molecular patterns of PAMT, which could be characterized as pro-inflammatory upon reaching liver. So all these pro-inflammatory signals, together with those from other sources, may trigger hepatic fibrosis and hence the NASH. Well, let's take a look at the information in the different animal models of NASH. So I was reminded of the ovarian observation that all animals are equal, but some are more equal. So the question really is, are the inflammatory processes in different animal models the same or different? So this is the question we'll try to address. Here are the two different NASH models in mice that we're going to describe for the next few 20 minutes or so. So in the first model, the hepatic steatosis and fibrosis are induced sequentially by high-fat diet, HFD, and carbon tetrachloride, CCl4. So this is a sequential induction model. In the second model, so-called MCD model, these two, steatosis and fibrosis, are actually induced simultaneously. So if you look at the histopathology score analysis, on the right panel, these two models are quite similar. There's, maybe with some quantitative differences. Okay, if you look at these features, they all include, both of them include, significant steatosis, inflammation, fibrosis, to a less extent, blooming. Okay, as represented by the color-coded bars in the graph on the right. So the color used in this in the subsequent presentation will be consistent. So blue being inflammation, green blooming, and likewise red is steatosis. The yellowish, orangish is the fibrosis. Okay, so histopathology scoring as used in clinical diagnosis of NASH alone cannot distinguish these two models, as shown on the right. Okay, so in other words, these two models, either they are equally good or equally useless. Okay, so it can be both. Okay, so we try to find out whether they are one is better or one is worse. So in terms of the clinical relevance. So when we did the gene expression profile analysis of the liver tissues from both models, the difference became apparent. The scatter plot on the left depicts the regulation of gene expression of overall 15,000 liver genes in these two models. Okay, the overall correlation efficiency is about 0.3. So these are represented by the open circles. Okay. In the background, and on top of this, so basically these two models, the overall profiling, gene transcription profiling is the correlation is pretty low. So if you look at the genes involved in inflammation, okay, the correlation drops to 0.05. So basically, the inflammation processes in these two models are not relevant, not related to each other at all. So now we want to find out, okay, so to what extent they're different. So in the first experiment, we tested two NASH compounds, obeticholic acid, OCA, and senosulfate, SEL, okay, and in both models. So in the high-fat CCl4 model, okay, obeticholic acid displayed significant efficacy in improving steatosis, fibrosis, whereas senosulfate were not efficacious. Okay, so this will be the top left panel. Okay, when we test these two compounds in the MCD model, the opposite was observed, namely the senosulfate was efficacious, whereas obeticholic acid was not. So we noted in both animal models, okay, the histopathology of inflammation was actually exacerbated by the compounds that show no efficacy. So these are the blue bars marked by white triangles in the diagram on the left. Okay, so the gene expression profiling further confirmed these differences. Okay, despite the signature gene expression changes attributed to obeticholic acid in both models. And I don't want to go through the detail, but shown on the right panel are the heat maps of the two subsets of genes involved in inflammation, lipid metabolism in these two models, and in response to obeticholic acid. Okay, so this slide summarizes the histopathology result of both compounds in both animal models. So the pharmacological effects of these two compounds in these two models are actually, I would describe as diametrically opposite. Okay, in 3 out of the 4 histopathology features, namely steatosis, inflammation, and fibrosis. As you can see, same compound in different models, the outcomes are different in the two models. Okay, so basically, I've shown evidence that inflammation, steatosis actually are different in these two models. Okay, so now let's take a look at the fibrosis. We want to see fibrosis, how different the fibrosis is. Okay, so again, the question is, which one is more equal? Okay, so next slide. Okay, so in this is the Cyrus red staining of the liver sliced from high-fat CCl4 model and the MCD model on top and bottom on the left panel. Okay, and the histopathology analysis reveals that fibrosis in these two models actually is different. Okay, if you characterize these fibrosis in these two models by the category used on the in the blue box in the table. Okay, so namely, the fibrosis in portal tract, paralural and bridging fibrosis, and chicken-wide, and overall structure. Okay, you can see that the high-fat plus CCL4 model. Okay, the fibrosis is structured and is nicely distributed in the relevant regions. And by the way, these features used to characterize fibrosis are clinically relevant. Whereas if you look at the MCD model, okay, so the relevance to the characters, okay, is much less, and you can describe the fibrosis in MCD model as amorphous. So. Okay, so the question. Okay, so here's another. Take a close look at it. Okay, so the previous slide. So this is the so-called macro or subanatomic structures. Okay, and if you look at the microstructure, okay, fibrosis, you will see in the high-fat plus CCL4 model, steatosis and fibrosis are co-localized, and the steatosis is represented by the white bubbles. Okay, these are fatty droplets. Okay. It's very clear that in this high-fat plus CCL4 model, these two are co-localized. Whereas in the MCD model, the fatty droplets, okay, they are more heterogeneous in terms of size. They're much bigger than what we saw in the high-fat CCL4 model. And there doesn't seem to be a very clean co-localization of steatosis with fibrosis in the MCD model. So now the question really is, can we quantify the difference of fibrosis in these two models? Okay, so before I get into the technology we used in addressing this question, so I would like to introduce my next speaker, Dr. Ho from HistoIndex, and he will present the stain-free technology, digital histopathology technology, and Dr. Ho, the floor is yours.
All right, thank you, Terming. I'm just going to make sure that I could be heard by everybody. If you're, Terming, you could hear me?
Yes, very clear.
Okay, very good. So thank you, Terming, and thank you, the WUSI, for extending the invitation for me to be able to come up here to share some insights on stain-free digital pathology using and being used in Nash preclinical studies. Also, thank you for the attendees, those who are here attending this webinar on the other side of the computer. Thank you. So I'm going to go ahead and see whether do I have control of the slides. I do. That's a really great thing. Disclaimer. Let's go on. So awesome animation, right? So today we have more than a third of the market share in AI-based digital pathology for drug R&D in liver. Both clinical and preclinical. The success of our laser-based imaging lies on the foundation of this technology known as two-photon microscopy. So what happened is that instead of one photon commonly used in confocal microscopy, we use two photons that have higher energy levels, resulting in a generation of second harmonic signal, or SHG, which can be used for high-resolution imaging of non-central symmetrical structures such as collagen fibers. And collagen fiber is a biomarker for fibrosis, which is the cause for about 40% of all deaths in the developed world today. So it's a very big deal. At the same time, together with the generation of SHG, transmitted signal from the two-photon emission we call 2PE, you can see it and on the diagram, on the cartoon itself, the red color ones, allows us to capture the information on the cells. So now you put all this state-of-the-art technology together and you compact it into a benchtop imaging system. The reliability of the Genesis imaging system, which can be seen on the left of the screen here, allows us to capture the same image every single time, regardless whether the scanning was performed in the United States, in US, in Europe, or in Asia. And together with our proprietary algorithm, we can then analyze these images at morphological levels in an automated and reproducible way. I'm aware that I cannot ask questions right now. I cannot take questions and answers right now, so I'll do it later. Now, all right, so today we will be discussing about AI-based stain-free digital pathology using the Genesis imaging system on solely NASH preclinical studies, looking at various animal models. So for HistoIndex in liver fibrosis alone, we have at least 60 research publications over the past 7 years using our Genesis SSG imaging platform. As you can see on the table, the SSG platform has been well validated in various NASH animal models covering a spectrum of genetic modified, be it diet-induced, chemical-induced, or even a combination. So some of these studies have been published or presented during conferences by our sponsors. And today I'll be sharing some of this data that are in the public domain. Okay, so in this slide, okay, so I do not have a laser pointer, so the audience got to follow my voice as I'm going to guide you through the different parts of the presentation within the same slide. So in this slide, we are comparing between stain-free using SSG versus stain using PSR, which stands for picosirus red, for assessing fibrosis. So before I proceed, just as a background, digital pathology and whole slide imaging, they have been around for about 20 years. But advances recently in photonic and computational technology, coupled with AI and machine learning, has brought a new wave of information and data into tissue diagnosis that could radically change how stains are used in the future. So if you look at the screen in front of you, you could see that the stain-free SSG TP images in the top row, right? It shows a 34-week child diet, followed by a 34-week high-fat diet, where you can see visually there's a huge increase in the green SSG signal that comes from the collagen fibers, and then subsequently a decrease in the SSG in the OCA treated group, right? And you're looking at it visually. And if you look below, you can see quantitatively in the graphs below that the increase and the reduction in the SSG numbers is signify fibrosis worsening and improvement, and they are about 100% more than the numbers you can see from the PSR stain, because if you look at the graph and the middle, that's a PSR stain, when the one on the left is the stain-free using SSG. So basically, in this 34-week study, it was shown that SSG is twice the sensitivity as compared to stains. All right. SSG is second harmonics generation. Again, let me just explain that. It's specific to type 1 and type 3 collagen, where you can visually see in the images in front of you that injury to the liver that causes a specific type of collagen to be produced in the early onset of the insult, and they are then laid down within the extracellular matrix. This could be seen and quantified by SSG in a 16-week high-fat diet model. We have even done SSG analysis in a 6-week choline-deficient high-fat diet NASH animal model to assess the worsening and improvement in fibrosis before and after treatment. So I hope that I've used the above two published studies to show that the stain-free SSG solution greatly surpasses existing stain-based technology in both the area of sensitivity and specificity. So what I'm going to do next is I'm going to use the next couple of slides to frame the use of the technology itself, especially in the advantages of both a heightened sensitivity and specificity in assessing the drug efficacy in NASH animal model. All right, let me shift to the next slide. Okay, there are many limitations with current pathology readings, and pathologists over a period of time will need new tools, and I think it's time that they need new tools right now. So this slide shows a good example of the solution provided by the SSG platform, where we compared pathologists' assessment of drug effect on the fibrosis using discrete categorical staging and HistoIndex, our platform using a SSG continuous way of measuring. You could see on the horizontal lines on the left, you follow that on the graph on the left right there, where the pathologist assessment having no change in fibrosis stages of the NASH animals before and after treatment is typically around 50% of the animals. Now, so question is whether this stable, no change of staging, is it because that the change could not be detected due to the pathology's discrete way of staging, or is it truly no change at all? So let me give you an example on the right. If you look at the graph on the right now, in this particular NASH phase 2 clinical trial, where the pathologist assessment having no change at all in the fibrosis stage of the subjects before and after treatment is about 55%, the green portion, right? You can see that. The actual percentage is only actually 16% of the subjects when we use a continuous measurement to assess the efficacy of the drug. So in both preclinical studies and even clinical trials using the platform, the SSG platform, not only shows the performance of the therapy objectively and consistently, but it also allows us to provide to the sponsors a dose-dependent efficacy of the drug. That's what we have seen and we have shown as well. So similar to the continuous measurement of fibrosis, now we're going to go straight into steatosis. The slide here expounds on how in conventional pathology's assessment of steatosis, in this case, showing a score of 1, which means the area involved by steatosis is between 5 to 33%, if you look at the table on top. 5 to 33% of the area of the entire tissue, and is a huge range based on just eyeballing of the affected steatotic area under the microscope. So you could just imagine right now that the lack of resolution to determine the efficacy of the drug, in this case the INT-767, which is a second generation of OCA, using such a wide range do that. And on the other hand, the continuous measurement of steatosis, and to be specific, is the macrovesicular steatosis using the SSG platform. It shows about 60% reduction after 8 weeks of intervention, while the pathologist's discrete score, it didn't show any change at all before and after treatment. It still remains as a score of 1. So not only could steatosis be measured continuously, but we could even plot a histogram, as you can see on the right, as you can see, and showing the spread of the number and size of the fat vacuoles in both the vehicle and the treatment groups. Okay, in a different NASH model, in this case is a diamond model, looking at the bar chart on the left, you could see that the limitation of the pathologist's categorical fibrosis staging is unable to show the dose-dependent anti-fibrotic efficacy of the drug. The chart shows both the low dose and high dose, and was shown at 0, basically. So, but in the continuous Q fibrosis value, the bar chart on the right, there's a trend of improvement in the Q fibrosis value with increasing dosage of the ST001 in this case. You could clearly see the step down trend of fibrosis improvement with increasing dosage. All right. So I hope I've shown in the past few slides convincing data on the advantages of the SSG continuous measurement of fibrosis and steatosis over that of pathology's categorical discrete staging or scoring. I've mentioned this previously. The platform itself, the SSG, not only shows the performance of the therapy objectively and consistently, but it also allows us to provide to the sponsors the dose-dependent efficacy of the drug. Okay, so in the next few slides, what I'm going to do is this. I will show examples of preclinical NASH studies using a very advanced AI-based analysis on the various zones of portal tract, parasinusoidal, or the CV, central vein, and the consequential changes in fibrosis and steatosis in these zones before and after treatment. So, for example, what happened to the changes in fibrosis as a result of changes in steatosis in these zones within the liver, and all these changes I fully quantify it in a continuous measurement. Okay, so I will need you to buckle your seatbelt, as we get in deeper into this. What I'm trying to next slide right here. What I'm trying to illustrate here in this slide is that the increase in the SSG measurement signify a worsening of fibrosis and statistically significant in a certain area of the liver. It known as a parasinusoidal region, or what we call zone 2. Now, diet-based NASH model like HFD, high-fat diet, typically has a low level of fibrosis, unless you put them a very long period of diet. The hepatotoxin CCL4, in this case, it acts like a positive control due to the chemical insult on the liver. And when you couple it with a high-fat diet, as shown in the yellow column right here, right now, Terming was saying that that's sequential induction. It causes an increase in production of collagen due to the activation of the quiescent hepatic stellate cells. So what happens is that your increase in collagen is predominantly very much in a parasinusoidal region. So you don't get to see that happening significantly in the rest of the region of the portal tract and central vein. You get to see most of them in the PS, which is a parasinusoidal region. And after treatment of OCA, so now we're talking about treatment, in the blue column, you look at the blue bar, the improvement in fibrosis is statistically significant in all three zones, whether it's a PS, PT, or CB. It shows the efficacy of the drug hitting across the entire liver. In short, fibrotic changes in the parasinusoidal region is a more sensitive indicator of both model induction, all right, sequential induction, as well as anti-fibrosis drug activity. All right. So if you dive deeper looking at this parasinusoidal, or what we call the PS region, to find out more, and looking at this slide here, this is SSG TPE image, is red and green, and that we acquire from the Genesis imaging of a mouse liver tissue. First, allow me to explain some new terminology right here. Other than quantifying the morphometric parameters, we also segment and quantify those collagen fibers that are around the fat vacuoles, that we label them as co-localized fibrosis. So these are fibers, and these are collagen fibers that intermingle with the fat vacuoles. We call them co-localized fibrosis. And conversely, those collagen fibers that are outside the statotic area, or all these fat vacuoles, we call them, or we label them as non-co-localized fibrosis. So within a tissue itself, you have both co-localized and non-co-localized. As you can see on the bar chart on the right, and starting with the CCL4 positive control group, is showing almost all the fibrosis in the PS region. The zone 2 is non-co-localized. Of course, because there's no fat vacuoles in it, they are all CCL4, so you don't get any co-localization fibrosis with statosis. But then even in the middle, HFD CCL4 animal group, that's where there's an introduction of high-fat diet, and you could see that the overall fibrosis, because of these, have worsened due to the fibrosis, and that accompanies with the introduction of statosis. This is known as co-localized fibrosis. So in purple, and what happened is that it basically shoot up. The amount of fibrosis just went sky high, rocket high. You can see that overall fibrosis just went real high. So the turquoise region, you add on with the purple region, you get almost a 200% and above. Now, with the treatment of OCA, there's an improvement in overall fibrosis with a 55% reduction in total collagen in zone 2. But if you look at just the co-localized fibrosis, the purple part, there's an 80% reduction in collagen accompanying the reduction in fat vacuoles. Therefore, fibrosis around fat vacuoles shows greater improvement as statosis improves in this particular region. Now, such information is a great asset for elucidating the mechanism of action of drugs, the MOA. You will expect the ratio of the co-localized versus non-co-localized fibrosis to be very different if let's say it's a MOA due to anti-fibrotic drug. For example, it's just attacking the anti-fibrosis part of it. So that's what happens. What I'm going to show you next is that what we have seen here and we have done in preclinical NASH, we have also demonstrated in NASH clinical trials. Now, as example shown here in two separate NASH phase 2 trials, phase 2B to be exact, that we have presented during AASLD 2019 and NASH Tech 2020, just this early this year. It shows how by co-localizing fibrosis and statosis in all three zones of PT, PS, and the CV, and it reveals the MOA of the drug, as I've explained previously, and is vital to evaluate the drug efficacy and how that information could be used in the design of the phase 3 trials, as well as in the sponsors' discussion with the various agencies. I mean, whether it's the FDA or EMA or NMPA. So I do not intend to spend too much time on this slide, as I'm mainly wanting just to draw points of comparison between the analysis done in preclinical studies and the clinical trials. I'm going to leave it as it is, and if there are any questions, we could always take it offline. Now, second last slide, this is showing the SHG and TP images of a biopsy from a non-human primate. So this is a monkey. It looks very similar to human biopsy with regards to the collagen profile and distribution of fat vacuoles in the various regions of the liver. We are in the midst of acquiring more data from our NHP studies on the advanced analysis of the various zones of PT and PS and CV and the consequential changes of fibrosis and steatosis in these zones before and after treatment. So the grand scheme of things is that we could compare the data from the NHP studies with the data from the clinical trials to ascertain the translatability aspect between non-human primate and the humans. All right, so that's what we do. So in summary, we have a highly sensitive and specific AI-based system to detect early and minute pathological changes in a measurable manner. We could quantify fibrosis and steatosis histologically in a continuous scale, and furthermore, deep dive into the analysis of the different zones of the liver tissue for data on the MOA of the drug. So this entire platform allows us to narrow the gap between NASH preclinical and clinical efficacy studies during the trials. With that, see, are there any more slides? Thank you very much. I'm going to pass the time right now to Terming. I think he has more to share. And Terming, it's all yours right now.
Yeah, thank you very much, Dr. Ho. Yeah, thank you very much for the nice introduction presentation on the technology. And in fact, we set up a collaboration with HistoIndex a while back, and trying to use HistoIndex technology to see how we quantify histopathology, particularly the fibrosis. Okay, so unlike the other digital or quantitative histopathology tools, comes with the package. Okay. The scanner, for example. Okay, the HistoIndex technology not only quantifies fibrosis and other features, but also it localize these features in different regions of the liver, okay, at the microstructure level. Okay, so namely, if you look at the diagram on the left, okay, so 3 key regions of the liver. So namely the portal tract, the zone 1 corresponding roughly to zone 1, parasinusoidal, zone 2, and the central vein. Okay, so the color coded, these regions are color coded pretty much. The panel on the right, that we will stick to the same color. So if you look at the distribution of fibrosis in high fat and CCL4 model, what you see is that the 2 regions, okay, the zone 1, zone 2, the portal tract and parasinusoidal regions, they have the most fibrosis in this model. And upon treatment of obeticholic acid, the reduction in these 2 regions are statistically significant, whereas the acetaminophen treatment did not seem to have any significant effect on the fibrosis in any of these 3 regions. And then if you look at the MCD model, okay, the overall fibrosis is lower than the high-fat plus CCL4 model. And whether it's obeticholic acid or acetaminophen, okay, there doesn't seem to be any significant efficacy on the fibrosis in these or none of the 3 regions. And by the way, the distribution of fibrosis in the MCD model is also quite different than what we see in the high-fat plus CCL4 model. Okay, so as Gillian just mentioned, this technology is actually quite powerful in terms of bridging the preclinical study with the clinical outcome. So I think this is a pretty good indication that at least the efficacy we saw with obeticholic acid in the high-fat plus CCL4 model is actually largely consistent with what we saw in the clinical trial, the outcome of the clinical trial. So what is the parasinusoidal space? And this is a very important space, anatomic structure. So I want to spend some time on this. Okay, so basically, sinusoids are the vesicle channels that receive blood from the terminal branches of hepatic arteries and portal veins at the periphery of the lubrus, because that's called the portal tract. Okay, it's on the zone 1, if you will, and deliver the blood to the central vein. which is zone 3. Okay, and this is a tiny. This is where the blood from the artery, the artery and the veins are mixed. Okay, and this the space, the parasinocidal space, also known as the space of dices, highlighted by the red labeling here. Okay, there's actually a tiny space between the endothelial cells lining the sinusoidal and the flanking plates of hepatocyte, and has been postulated that the resident macrophage, also known in the sinusoidal, also known as the Kupffer cells, are activated by toxic lipid species, oxidative stress, and pathogen-associated molecular patterns such as LPS, and they secrete TNF-alpha and other pro-inflammatory molecules, and that inside the parasital space. Okay, and the signal goes to the parasital space, and it is the stellar cells in the parasital space, upon activation by such signals, secretes extracellular matrix, among them as the collagen, and hence the initiation of the fibrosis. So really, this is the tiny little space between the sinusoid and the plates of hepatocyte. This is where the fibrosis started. Okay, so if you look at our results, okay, so it's very clear that the parasital space is the major region, one of the major regions where the fibrosis is distributed in the high-fat plus CCL4 model, and likewise the portal tract region is also highly distributed in terms of fibrosis. Okay, and upon treatment of CCL4, there's a statistically significant drop in these two regions, basically zone 1, zone 2. Okay, whereas if you look at the serum subito, okay, you don't see this improvement. And in the MCD model, we don't see this distinctive distribution of fibrosis in the parasinusoidal or the portal tract region, whereas the three regions are more or less evenly distributed in terms of fibrosis. Okay, so the antifibrotic. Let me go to that. So here's another view if you look at it. Okay, so we're looking at the macro structure. Okay, so excuse me. So we're looking at the fibrosis. Okay, shown. I mean, okay. What is the the portal tract region? Okay, so this is basically the region at the periphery of the lubra. Okay, and that's where the fibrosis also happens, and this is where the bridging fibrosis between the neighboring portal tract is initiated. And as you can see that in this slide, okay, the green arrows indicate the portal tract region that was heavy fibrosis, and the blue arrows indicates the bridging fibrosis seen between the neighboring portal tract. And upon treatment of obeticholic acid, both types of fibrosis are significantly diminished. Okay, so our conclusion from this analysis is that the CCL4 model has the characteristic, if you will, Nash-like fibrosis, and it is the this efficacy of obeticholic acid in these two particular regions of the animal model. We believe lead us to believe that this model is more clinically relevant. Okay, let's move to the next slide. As Gillian mentioned, the co-localization of steatosis and the fibrosis is an important feature of fibrosis of Nash in the clinical trial. Okay, so as also mentioned early on, the co-localization of the steatosis and the fibrosis in this high-fat and CCL4 model, as you can see, upon obeticholic acid treatment, Okay, the steatosis associated, the fibrosis associated with steatosis, okay, the purple bars, okay, actually is significantly reduced upon treatment. Okay, and likewise, the steatosis is also significantly reduced. Okay, so what about the turquoise bar? Okay, the light blue color. Okay, this actually we know that. Okay, so this is actually this fibrosis induced by CCL4. So in other words, okay, the fibrosis we saw in the high fat and CCL4 model, the extra level, okay, is actually induced by the combination of high fat diet plus the CCL4. So it is that particular part of the fibrosis is a target of the obeticholic acid, and which is largely consistent with what we saw in the clinic. So what other evidence we have to support our assessment that this model is more clinically relevant? Okay, so I'm going to show you just one more slide, and this basically is we took the I think 4 of the 5 clinical compounds in the phase 3 and 1 in phase 2. Run them through the animal model. As you can see, obeticholic acid displays great efficacy, and likewise, the magical compound is also efficacious in this model, whereas tenosirbitol and anifribinol, okay, and are not does not show significant efficacy. In particular, the anifribinol. Okay, so if you look at this, if you look at the result, okay, with this particular dosing, okay, the fibrosis is significantly reduced. Okay, however, in this particular dose, okay, fibrosis is reduced. Okay, at this particular dose, okay, the steatosis is actually increased. Okay, so it's kind of very intriguing. Okay, very intriguing. Okay, so with that, it's very hard to call that efficacy. Okay, but all in all, so we believe the efficacy of our model, the outcome is largely consistent with the phase 3 or phase 2 clinical trial. So basically, I hope I have presented evidence that these 2 models we discussed, MCD and the high fat plus CCL4 models in this webinar. Okay, they're not the same. Okay, and the high fat plus CCL4 model is probably a better representation of NASH, and our analysis using quantitative or digital histopathology by a histoindex, okay, demonstrates that the fibrosis specific to the high fat plus CCL4 model is NASH-like, and the fibrosis is also associated with steatosis in this model, and both of which are the therapeutic targets of obeticholic acid. So obeticholic acid by far is the most advanced NASH compound, despite the recent unfavorable CIL from FDA. Anyway, so as in the fibrosis, steatosis, as in the statistics, okay, right? So as I mentioned very early on, all models are wrong, but some are useful. So I hope that I have demonstrated that the utility of the high-fat plus CCL4 model. And on behalf of Dr. Ho, I'd like to thank the audience for your participation, for your time. And I also want to thank the team, Dejan and the US marketing team for making this possible. And we're going to take some questions.
Tommy, there are some questions regarding about the models, but I think I will take those questions first regarding about SSG, the platform. Is that okay with you?
Sure, yeah, go ahead, yeah, please.
Yeah, okay, so I'm gonna just, the questions between the CCL4 high-fat diet model is intermingled with some of my questions, so I'm gonna sieve it out. First of all, for Patrick, SSG study is SSG certified by FDA for use in NASH clinical trials. In NASH clinical trials, you do not need a certification by FDA. We are actually working on a submission to FDA rather for the NASH clinical diagnostics. So we are being used in trials in basically 1/3 of all trials, phase 2 and phase 3 trials in NASH today uses SSG. Okay, I'm going to move on. That's next one. Could you, let me see. All right, there's a good one from MSD, Assad. When you say co-localized, what does it mean? How many micron of fibrosis and statosis is called co-localized? So the algorithm recognized co-localization of fibrosis and statosis as within the same region, within the same vicinity. So the fibrosis, which the collagen fibers itself, are actually within the sinusoidal space where the fat vacuoles are being located. So that's co-localized. So I hope that answers in terms of what you want to address. Okay, so let me see. I think there's a one which there's a pretty good questions. That's Bahasini, Ridgeline Therapeutic. Okay, how are statosis and fibrosis continuously monitored using Genesis? Is this via a in vivo scanning? It's not in vivo. It is ex vivo, so it is still histology based. When we talk about continuous, it is a continuous way of measuring the both fibrosis and statosis as compared to pathologist's way of discretely categorizing them in terms of their severity. So normally in fibrosis, pathologists use F0 to F4. That's the Nash CRM pathologist scoring staging method, and statosis is S0, S3. So these are 0 and 1 and 2 and 3. Whereas when we look at continuous measurement, it is basically 1.25 to 2.63. Basically it is continuous. There's no discrete scoring when it comes to that. Yes, I'm going to pass the time over to Thurmi right now because he should take some and let me have some time to look at the rest of the questions. Yeah. Okay.
Well, thank you very much. Thank you very much, Dr. Ho. Yeah.
Right.
So I have a lot of questions here. So if the ones are already answered in the slides, I will skip them. Okay. So the first question comes from Segni with regard to the bile-acid. If we have done an assay, we have not. Okay. So the 3D, how the Christine from CBQ asking how these animal models stack up against the 3D. We actually have the in-house 3D, what do you call it, the precision cut liver slice, okay, in process, it's setting up. And we don't know, I mean, we haven't done the direct head-to-head comparison yet, okay. So next question comes from Xing Yan and Genevieve, and asking how this CCL4 plus a high-fat diet model compare with CDAA, FDA, a high-fat. As far as I know, we have not done the direct comparison. Okay, my understanding is that CDAA, the high-fat model, the fibrosis in that model is totally different. Okay, so it's more like the fibrosis in that model more resembles the MCD model. Okay, so the fibrosis are different. Statosis in a way, there is some overlapping. Okay, so that's as I can. We have Alexandra elaborating on the timing, posing on the. Okay, so this is a pretty long question. So, okay, so we actually just. If you're asking a technical detail, I can give you the answer afterwards. Okay, this is a pretty actually long question. Okay, we're gonna skip. If I have time, I'll get back to you. Okay, so diet-based model like clinical relevant. Yes, the second question comes from Ingrid. Okay, the next question, research. Cardiometric, and yeah, that's a good question. Okay, so we are actually setting up we run the MLN model. Okay, so we know that, and the fibrosis, it's the timing of the fibrosis, how long it takes to induce fibrosis. As you know, we're running preclinical study. We want to have a reasonable timeline, okay, in terms of running the animal model. So right now, the animal models we have, the whole experiment period, okay, is about 20 weeks or within 20 weeks. And if you if you take out the time involved the induction of DIO, okay, so the actual experiment is 4 weeks in the CCL4 plus high-fat model. Okay, so whereas in this model, it will take considerable longer time to induce fibrosis. Okay, Raham, I think. Alexandra from Lily regarding. That's a very good co-localization of steatosis. Any thoughts? Actually, very good question. Yeah. So there's another. That's a very good. I have to think about it hard. Okay, so we can discuss offline. Okay, so yeah. Slide 36. This is the histopathology, done by the pathologist. Okay. However, we do have the histoindex quantitative data on these compounds, so we can share this another time. Okay. So basically, what we saw with the histoindex technology is consistent with the histopathology saw. Okay. So namely, if you look at the anfibrino, okay, right, the co-localization, the problem is the co-localization, okay, of the efficacy of between fibrosis and steatosis. Can we get the slides? If you send me an email, we can discuss this, okay? How much? That question I don't know. How much semaglutide is used to handling Nash? I actually, I'm sure the data is somewhere there, okay, but I really don't have this on top of my head.
It's semaglutide ones, yeah, right? So,
yeah. So Jennifer's colonized fibrosis, non-colonized fibrosis. I don't have the data, but I don't know if Gideon you have the data. This is the question from Jennifer Chen, okay, from UCSF.
Do you have right? Let me take a look at it. Well, I have so many questions that I have no time to actually look into it. But do you have the data on co-localizing fibrosis and non-co-localizing fibrosis with choline-deficient high-fat diet? Yes, I believe we have. We have done that particular model. We will have to look it up, Jennifer, for that. Some of you are asking about STEM model as well. I think STEM model, I saw a STEM model. We did STEM model too. I think it was asking us to comment about it. STEM model, offhand I couldn't tell you about that particular characteristics of the STEM model, but I think if you get in contact with my team itself through the QR code thing, I could get you the answer. So the slides are stain-free. So in a sense, when we scan it under the Genesis imaging system using SSG, these are non-stain slides. So after which you could actually use the slides back to do whatever you want to do. You can do staining post. The analysis post after we have finished scanning it. The slides are still reusable. You could just stain it, and you could actually be able to merge it with the exact image of the SSG and the 2PE images together. So that's one thing. NGS data in slide 9, I guess that's for you, Deming. Okay.
Yeah, so I have a question from Rajiv and asking, the mechanism of butyrate acid in the high-fat model. Okay, so we're actually looking into that in terms of the fat composition, after treatment. Okay, so we're doing that. We're also actively pursuing the fat, if you will, the metamorphosis of lipid species. Okay, after prolonged induction. Okay, so I want to find out, how the lipid profile has changed in the model and upon the treatment of butyrate acid. So we have some understanding of this in the future. Okay, so index, what is the drug treatment? Cup model, very good. We actually have not looked specifically at the Kupf cells. Okay, so I think this is something we want to look into, and this is something I actually want to do with the ex vivo precision cut slice liver slice. Okay, this is something we want to do with this, and also we're exploring the co-mixture, the co-culture of hepatocytes with Kupf cells and stellar cells. Okay, so this is something we were looking into this. Okay, so
thank you, Deming. Thank you, Deming and Gideon. I just realized that we're up on the hour now. And so we will. This will be the end of our presentation. We will respond to any questions not answered offline. This will conclude our webinar. Thank you, participants, for being a great audience. Everyone, please visit wuxioptic.com for more information, and have a great day.
Thank you.
Thank you.
Thank you. Thank you.
Thank you. Yeah.